Welcome to the NIH Cloud Module, a cloud-based learning module for biomarker discovery. This module was developed by the Rhode Island INBRE Molecular Informatics Core as part of an NIGMS administrative supplement to the RI INBRE grant. Rhode Island INBRE is based in the College of Pharmacy at the University of Rhode Island. Biomarker discovery is a common task in biomedical research. A biomarker is a biological entity that indicates a change in state, often associated with disease or injury. Identifying practical biomarkers aids in the diagnosis, prediction, and treatment of disease. However, the process of identifying and assessing the clinical utility of biomarkers is a technical challenge. Just as challenging is communicating scientific results between different parties. For the past 2 decades, omics data sets such as proteomics and transcriptomics have been targeted for high-throughput biomarker discovery. For a bioinformatician, omics datasets provide a comprehensive systems-level view of molecular changes occurring due to disease progression, disease treatment, or across diverse populations. This view provides deep insights into the molecular mechanisms of these changes and aids in large-scale biomarker discovery. For the clinician, however, the primary focus is on patient outcomes and biomarker utility. While a broad view of the system might be informative, a practical clinical biomarker must be cheap, easy to measure, and diagnostically useful. Bridging this communication divide is important if the biomarkers identified through experimental analysis are to be translated into practical clinical applications. For this model, we are using as our case study a rat renal ischemia reperfusion injury model using biomarker and proteomic data provided by Dr. Nissan Gonam, Department of Biomedical and Pharmaceutical Sciences, College of Pharmacy, University of Rhode Island. IRI is a critical problem in organ transplants which can lead to significant damage or failure of the donor kidney. Ischemia occurs when the kidney is removed from the host body and temporarily deprived of oxygen. As cellular ATP stores drop, mitochondrial damage occurs which can lead to cell death. When the kidney is reattached to the host, the sudden influx of oxygenated blood, called reperfusion, results in the formation of reactive oxygen species which leads to further cell damage, inflammation, and eventually cell death. At this time, there is no approved pharmaceutical treatment for mitigating the effects of IRI. But Dr. Gonam's lab is studying potential treatments using repurposed FDA-approved drugs. Renal IRI was chosen as the case study for this module because of well-known kidney damage biomarkers which can be used as references for assessing biomarkers identified by proteomic analysis. The module consists of 5 core submodules and 4 optional supplementary submodules. Core modules include Introduction to Biomarkers, Rat Renal IRI Case Study, Linear and Logistic Regression for Comparison of Quantitative Biomarkers, Exploratory Analysis of Proteomics IRI Data, and Identification of IRI Biomarkers from Proteomic Data. Optional submodules include Introduction to R Data Structures, Introduction to Linear Models, Principles of Exploratory Analysis, and Machine Learning Methods for Biomarker Discovery. This module is currently hosted on the NIH Cloud Lab through Google Cloud Platform. The submodules are written using the R programming language and implemented using Jupyter Notebooks. This project was funded as an administrative supplement to the RI INBRE grant P20GM103430. The funding was provided by NIGMS and administered by Dr. Lakshmi Madhukamali. Dr. Bangsup Cho serves as the program director of RI INBRE. The module was written by Dr. Christopher Hemme, director of the RI INBRE Molecular Informatics Core. Development and implementation of the module was assisted by a support team from Google and Deloitte, consisting of Laura Beaudry, Zelenka Yousafzai, Ross Campbell, Alan Kim, Daniel Pan, and Marsha Price. The rat renal IRI data was provided by Dr. Nissan Gonam with her students, Dr. Maywen Dang and Dr. Joyce Ho.